Okay, we will continue with the third part of protein, which is protein classification. So proteins are classified into three main groups, which are simple protein, conjugated proteins, and derived proteins. Now, what is simple protein? Simple protein is when protein hydrolyzed, the product of protein hydrolysis are only amino acids. And example of simple proteins are albumin, globulin, glutein, prolamins, scleroproteins, histones, and also protamines. Now, what is conjugated proteins? Conjugated proteins are when the amino acid part of a protein combined with a non-protein material such as lipid, nucleic acid, or carbohydrate. And the major example of conjugated proteins include phosphoprotein. So based on the name phosphoproteins, the non-protein material is phosphate. Second is lipoproteins. So the non-protein material is lipid. The third one is nucleoproteins. So the non-protein material will be nucleic acid. The fourth one is glycoproteins, and the non-protein material would be carbohydrate. And the last one is chromoproteins. So the non-protein material is colored prosthetic groups, and that includes example like hemoglobin, which give our blood color red, chlorophyll, okay, which give the green color of plants. Another example is myoglobin, okay, and many other colored group. So the third classification of protein is derived proteins. So what is derived protein? So this type of protein are obtained by chemical or enzymatic method, and this is further divided into either primary or secondary derivatives. So what is primary derivatives of a derived protein? So these are the slightly modified protein which are insoluble in water. So the example of these primary derivatives is rennet coagulated casein. So rennet is an enzyme obtained from the cow, and it is used to coagulate casein, okay, from milk. Okay. So the second type of derivative is secondary derivatives. Okay, so this type of derivatives are extensively changed, which include protease, peptone, and also peptide. The difference between secondary the difference between group like protease, peptone, and peptide in secondary derivatives are in terms of their size and solubility. All of secondary derivatives are soluble in water, unlike primary derivative, which is insoluble. Secondary derivatives are not coagulated by heat. And lastly, protease can be precipitated using saturated ammonium sulfate solution. Next, we will look at protein hydrolysis. So hydrolysis means you are breaking down or the peptide bond which link one amino acid to another. Now, how we can hydrolyze protein? We can use either acid or base or water and heat. Now, hydrolysis of protein will give us smaller peptides and amino acids. This hydrolysis is also similar to digestion of protein using enzyme. So when we digest protein using enzyme, we are breaking down the peptide bond also. So protein hydrolysis occur in cell to give amino acid or to provide amino acid for further use in the synthesis of other protein and tissues. So this is example of hydrolysis of a dipeptide bond. So dipeptide bond means you have two amino acid joined with a peptide bond. Okay, and you break the peptide bond either using acid or base in the presence of water and also heat. Okay, so you break it. Okay, you break the peptide bond, giving the individual amino acid. So what are the hydrolysis when these alanine serine valine peptides are hydrolyzed? So therefore you should get alanine, serine and valine individual amino acid. Next, we move on to denaturation. So what is denaturation? It's a phenomenon that involves transformation of a well-defined folded structure of a protein formed under physiological conditions to an unfolded state under non-physiological conditions. So what does this mean? It means that you have a folded structure initially of a protein, it becomes unfolded. Similar like you fold clothes. Okay, after folding the cloth, and then you unfold it because you want to use the cloth, so it becomes unfolded. Okay, so that's one example. Okay, simple example of folded to unfolded. Okay, so in term of protein, it means you are changing the structure of the protein, okay, without disturbing the initial backbone of the peptide bond. Okay, so you have studied the primary structure, secondary structure, tertiary structure and quaternary structure. So unfolding means the high the structure, for example, initially is quaternary, so when it become unfolded it becomes tertiary and secondary. Okay, but the initial backbone does not break apart. Okay, so that's what it means by denaturation. Now, what are the factors that contribute to the denaturation of proteins? The factors are heat, salt, pH, and also mechanical action. So this figure shows the protein denaturation, okay, from the folded state to unfolded state. So notice that the the backbone remain the same here. Okay, from folded becomes unfolded. Okay, tertiary to secondary, and then primary. So the backbone still remain intact, no cleavage of the backbone. Denaturation will lead to loss of function. Okay, loss of function. And of course, loss of structure due to the unfolding. We will cover in terms of loss of function later on. Okay, so this is example of factor which contribute to protein denaturation. So the protein will denature when undergo heating, okay, whipping, or addition of acid or salt. Okay. So from folded state to unfolded. Okay, so that is denaturation. Okay, so we know that the protein becomes unfolded when it undergo denaturation. So what are the loss of function? Okay, for example, here, when you cook egg, okay, initially the egg is in liquid form, and once you apply heat, what happen is the physical property of the egg will change and it become solid. Okay, solid. So that is loss of function. Okay, from liquid, okay, to solid. Okay, due to coagulation. Okay, too long of cooking can lead to rubbery properties. Okay, so this is another example of protein denaturation, or the unfolding structure of protein. Okay, so what makes them become rubbery? Okay, it becomes rubbery when the protein bonds with another protein, and at the same time it is trapping water in between them. Okay, now denaturation can becomes partially reversible. Okay, for example, when it undergo mechanical action. Okay, in this figure here shows when when an egg is whipped, okay, whip or whisk, so it will form foam, okay, due to incorporation of air during the whisking. Okay, and when you left it to stand, this foam will collapse back into liquid. Okay, so that is example of partially reversible. Partially reversible means from foam becomes back to liquid. So these are other type of denaturant for protein. Okay, besides high temperature, low temperature can also denature protein. Okay, means when you put the protein under low temperature, it becomes denatured. Okay, for example, milk. When you put milk in fridge, okay, it will separate, forming the watery part and also solid part. Okay, the third denaturation here is heavy metal. So this heavy metal such as lead, cadmium will react with the disulfide bond of the side chain, which form solid. Okay, the next one is oxygen radical, or any ionizing radiation. This will permanently damage protein structure. Last one is cheiotropes, for example urea, guanidine hydrochloride. These these cheiotropes are highly denaturing agent and often used in protein folding studies. Okay, this is guanidinium chloride and urea. Okay, common denaturing agents. So this graph shows the percentage of protein the percentage of unfolding protein, okay, versus denaturing. Okay, so initially the protein is in folded state. Okay, so when the concentration of denaturant is increased, okay, the percentage of unfolding protein becomes increased too. Okay, initially quite packed here, okay, due to the folding structure of the protein, for example, quaternary, so it becomes unfolded as the denaturant concentration is increased, so it becomes unfolded. So these are the application of denaturation. For example, boiling of an egg. Okay, so that's one example of denaturation. Wiping the skin with alcohol swab for injection. Now there's a purpose or the reason. Before you get an injection, the skin is swabbed with alcohol because you want to ensure that there's no bacteria present, which will affect the injection process. Okay, so bacteria mostly are made of protein, so when alcohol swab is applied, it will denature the bacteria itself. Okay, so it will ensure there's no bacterial infection when you undergo injection. The third one is cooking. There's reason why you must cook raw food before eaten, okay, to destroy organism like E. coli. Fourth one is autoclave. Okay, autoclave is used to sterilize instrument, means to kill every possible source of contamination. Okay, and lastly, milk heated to make yogurt. So that is one example of denaturation. Okay, to prepare yogurt, milk is heated.